So I'd like to get started with this. As some of you may or may not know, next week is quite important. I'm originally from Argentina, and next week is quite important. The World Cup's starting. I know that for many people in the U.S. they don't care about this, but this is an event that happens every 4 years, and it's pretty amazing for mostly everyone. So this mostly everyone else. So this guy over there is Messi. He's arguably one of the best players in the world, and what he can do is quite amazing. And it involves vision and motor commands and motor preparation and motor skills and whatnot. And we can talk about his legs and his speed and whatnot, but I would argue that the magic really happens in his brain. So the brain is the one that has to have the vision, the decision-making, the rapid processing, and ultimately the skills to orchestrate all these type of motor outputs. And I'd like to contrast that with this type of video. I don't know if anyone has seen this, which is, this is actually a little bit outdated. I think this is 2011. I couldn't find a more recent version. This is the World Cup in terms of robot soccer. So this is where we are nowadays in terms of what robots can do in terms of playing soccer. So. So I'm going to advance a little bit. Okay. Anyway, so the speaker is sort of making it very exciting by shouting and saying all sorts of things. But this is sort of state of the art, at least in 2011.
What's the guy doing behind him?
What's that?
Oh, the guy's there to catch the robot and get the balls over?
Yeah. Yes. So I think arguably, I'm not trying to argue that playing soccer is the pinnacle of intelligence by any standards, but I think that this argues that the brain is doing a lot of magical things. I would like to understand what's happening inside neurons and circuits of neurons that can orchestrate the difference between someone like Messi or this. So this is, of course, another example. Again, good luck trying to implement this with robots these days. Okay, so let me close this and get back to the presentation. Was there a question, Bud, or comment? No? You're from Brazil? All right. Okay. So we know that the brain can solve many of these problems that we ascribe to intelligence, and arguably the brain is a product of millions of years of evolution, and that has led to interesting solutions to complicated computational problems. So by virtue of understanding how neurons and circuits of neurons solve these problems, we may understand biology, but also be able to instrument some of these ideas into computational algorithms. Biologically plausible or brain-inspired algorithms are shown not the only possible ways of solving these problems. One can engineer a lot of hacks to many of the questions that have to do with intelligence, but we may be able to capitalize on the one system that we know can solve a lot of problems that relate to intelligence. So some of the many features that brains have and which I don't know why this is.
Is a loose connection, perhaps?
Okay, I think if I hold it all the time, then it will work. No.
There was a comment on the whiteboard that you needed a specific resolution. Maybe you need to change the resolution.
Do you think this has to do with the resolution?
somebody erased it, but on the whiteboard it was written on the top line. the magical resolution.
This is a magical resolution? All right. Okay.
Yeah, it's one of the standards.
Yeah, no, I don't remember seeing that. Let me try. Does anyone remember the magic number?
I think it's 9. Well, maybe by 7
Cool. Okay.
The resolution of the presentation may be different than the resolution of the screen that's not being presented.
Okay. Let me quickly see if I can try another adapter and see if that's the problem. Just a second. Okay, well, here's a claim. I was going to say that one of the major aspects of computations in the brain is fault tolerance. And this is a perfect example of how computers are not there yet. And also the notion that the brain can learn and adapt to new situations. So maybe I can hope that you will adapt to this annoying jitter over the course of the next several slides, unless anyone has a better idea of how to, or wants to play with this. Okay, so here are a couple of things that I particularly admire about brains and circuits of neurons that I think most of you would agree are not quite there yet in terms of computers and robots. So we have hardware and software that work for many decades. I wonder whether any of these computers that we have today will be available and useful and working in 5 years, let alone 7 decades. We have parallel computation with serial bottlenecks. We have reprogrammable architectures. The same piece of cortex can be used for very different tasks. We can do single-shot learning. We can discover structure in data. Fault tolerance is of course, a major aspect of computations in brains. You may hit your head or drink alcohol or lots of other ways of trying to damage neurons, and still we can function quite well afterwards. Try that with your computer or your iPhone, and they are not so resilient to damage. And one of the major things that we've talked about already and will continue to talk about in the next couple of days is the robustness to sensory transformations, illustrated, for example, in the context of visual recognition, where you want to be able to recognize an object in spite of the myriad of different presentations that object can have in terms of different scales, positions, angles, and so on. And also you have interaction among many components and integration of different systems, which happens almost instantaneously. And this is also a major theme in engineering artificial systems. Okay. So in the context of CBMM, the Center for Brain, Minds, and Machines, we're very interested in studying neural circuits to try to understand biology, but also to try to interact with many of the other efforts, and to be able to explore the type of high-level phenomena that we describe in Thrust 1, 3, and 4, that is in the context of social intelligence, in terms of integrating intelligence and the development of intelligence, trying to understand how those are instituted in terms of brains and circuits. I'll try to argue and give you a few examples that this is a golden age to study neural circuits. We have unprecedented capability to interrogate and stimulate and interact with brains in ways that we never had before. And that gives us the opportunity to really be able to test complex computational theories and also be able to inform and constrain those theories from the point of view of neuroscience. So we can rigorously test some of the theories that happen that will happen in class 5, and some of the empirical findings from biology can be readily translated into code. So I put a couple of slides, mostly so that you have them later. These are some books on neuroscience and computational neuroscience. This is a somewhat obscure one, not one of the most famous ones, but one that I like very much, that gives a very rigorous introduction to computational neuroscience and the sort of computational models and ideas that I'm going to be talking about, and other people will be talking about today. So let me now give a very brief introduction to neuroscience, and again, I know this will be deja vu for some of you, just so everybody's on the same page in terms of some basic jargon. And again, feel free to interrupt with questions. Here's a schematic of the brain. The brain can be divided into different lobes: the occipital lobe, the frontal lobe, the temporal, and the parietal lobe. And from the early days in which people tried to study brains, we came to the realization that there is a certain degree of specialization. That is not, the whole brain is not completely uniform. And one of the most impressive examples of this is Broca's area, an area that's particularly important in the generation of language. And this was discovered a long time ago. It's particularly localized for most right-handed people on the left hemisphere, and damage to this area renders people essentially aphasic and unable to produce. So this is not a whole uniform mass of neurons, but there's specialized subdivisions of neurons involved in different types of functions. So much so that people like Brodmann spent a lot of time looking at the cytoarchitectonic structure and trying to divide the brain into a lot of different areas depending on the specific morphological properties of neurons. To some extent, the story in neuroscience begins with this guy over here, a Spaniard called Santiago Ramón y Cajal, who spent his time mostly drawing, looking at stainings of neurons and doing drawings, and had a very powerful intuition to understand some of the basic principles, or to suggest some of the basic principles that govern the function of neurons. So based on those drawings, he realized that neurons are cells, as opposed to what his mentor and competitor was saying. Golgi thought that the brain was a continuum and was not divided into individual cells. And he insisted, and largely he was correct, that the brain is composed of specific cells and neurons that have a nucleus, that have dendrites that provide input to the nucleus, and then an axon that provides that then sends information to other neurons. This basic flow of information is largely correct. That's essentially the way we understand flow of information in neurons. And a lot of the magic computations in our brains happen in exactly how information is integrated from multiple dendrites into a soma to decide whether the neuron will fire an action potential that will propagate the action. So I would argue that the brain is the most complex system ever studied. It's more complex than, say, studying the universe, in the sense that every one of those neurons is interacting with a lot of other neurons in very complex ways. I haven't been able to solve this, I don't know why it keeps shaking. Yeah, I tried to post a that person. I tried two or three. That's okay. So if you can stay here, I'm just Hi.
If it's not too much of a distraction, if we just wait till the break, I'll come back and I'll try and do it.
Okay, all right. So there are 10 to the 11 neurons approximately in the human brain. The retina alone has 10 to the 6 neurons. Each neuron connects to about 1,000 to 10,000 other neurons, and in turn receives input from about 1,000 to 10,000 other neurons. And most of the magic happens in the part of the brain that's referred to as the cerebral cortex, which is about 3 millimeters thick, and involves most of the type of computations that allow us to see, to hear all the sensory modalities, as well as planning and orchestrate motor. There's a wide diversity of different types of neurons, and there's a whole community of people in neuroscience trying to characterize different types of neurons in terms of their shapes, in terms of their particular patterns of genes that are expressed. And by and large, I would argue that we know relatively little about the specific function of different types of neurons and how they contribute to different types of computations. One exception to that is the notion that there are some types of neurons that are excitatory and other neurons that are inhibitory, and that plays a central role in many computational models. Beyond that, many computational models do not really get into the details of the different types of excitatory, inhibitory neurons and how they facilitate different types of computations. If we dig a little bit deeper in resolution within a neuron, there is a bilipid membrane that has channels. The flow of ions through those channels is ultimately what dictates the flow of information. And one can essentially describe a neuron by an equivalent electrical circuit that takes this form. That has a conductivity for each of the main ions that pass through the membrane, including sodium and potassium. And for each of those, one can write a very simple Ohm-like equation that at the end describes relatively well how ions flow and voltage is integrated in the neuron. As the information flows through the neuron and needs to be passed on to another neuron, the main way of communication between neurons is via chemical synapses, where if you have a nerve terminal from a presynaptic neuron, there's a release of neurotransmitters that then open ion channels in the postsynaptic cell. So later on, we'll talk about the basic models by which circuits of neurons instrument computations, and we'll write equations of this flavor, where X represents the activity of a set of neurons, W represents a set of weights, and then there's a nonlinearity, and this is integrated to create a given output. To a coarse first approximation, the way most of us think about in terms of biology, in terms of what's happening here, is that these weights, which represent the impact of a given neuron onto its postsynaptic target, have to do with what's happening here at the synapse. For a given amount of activity coming on the presynaptic neuron, there will be a different degree of influence or impact on the postsynaptic neuron. And that's governed by these weights, W in here. So this is an electron microscopy high resolution view of the very, very small distance that happens in between the pre and postsynaptic neuron. And that's essential to ensure the specificity of the communication between a neuron and the next. So I'd like to discuss now some very basic aspects of how we build models of individual neurons, at least some of the most basic type of models. So there's a wide variety of models that people have used to describe the function of individual neurons, starting with filter-like operations, where you take an input and that's filtered to produce an output, onto the type of integrated and power circuits that I will describe in a second, what people refer to as Hodgkin-Huxley units, multiple compartment models, and then models that include every single channel in every single dendrite of neurons. So as we go along this line here, we have more biological accuracy. There is a lack of analytical solutions. So here we have some hope of being able to do math to describe the neuron. Here, we largely lose that ability because we are bogged down with simulations and a lot of complex non-linear. So the computational complexity also increases in this direction. I'm not going to say much about this, but this is just to give you an idea of the basic way of communicating information in a passive way within a neuron. So without the propagation of action potentials, people describe an axon, or most typically a dendrite, as a basic cable. And one can use all the basic equations from physics to describe the propagation of current in a cable of this form. So this is Ohm's law describing the amount of current in between these 2 points. This is the voltage at this point, this is the voltage at this point, and this is the resistivity of this cable. So one can write what's called the cable equation that describes how voltage changes as a function of space and as a function of time. So by and large, within dendrites, voltage is propagated in a passive way, so there's no regeneration of the signals in an active way, and that means that voltage decays quite rapidly, and this is not the preferred way of communication for signals over very long distances. So you cannot really send signals to move your legs from your brain in this way. You need action potentials, you need the active propagation of signals. But within short distances, in particular within dendrites, this is a semi-accurate description of how voltage changes over space and time. So if we want to think about how to model individual neurons, again, people use a wide variety of different models with different degrees of accuracy and resolution. I'm trying to illustrate those models with this simple diagram here. So one of the, particularly in the context of most of what we're going to be discussing today, the emphasis is in trying to understand what happens at the level of complex circuits. And most of the models assume assume that a neuron is a single compartment, meaning that all the computation happens in one place, and ignore all the intrinsic complexities that happen at the level of dendrites as well as axons. If we go in the opposite direction, there are people who try to reconstruct the exact shape of a given neuron, every single twist in every single dendrite, every single axon, and ask what's the impact of this type of architecture in terms of computation. In between these two extremes are what people call multiple compartmental models, which means that there are multiple compartments. For example, here there's an axon, a soma, and 3 different dendrites, and one can start subdividing that more and more and more. And in general, the central question in neuroscience is what's the right level of abstraction that we need depending on the particular problem and question that we're interested in. It's not obvious to me or to anyone that more biological complexity is necessarily better. So in spite of the fact that there's a lot of people who are interested in characterizing with ever increasing degree of precision what happens in smaller and smaller compartments, from the point of view of understanding the ultimate output, input-output relationships for a given neuron, it's not obvious that we always need to have more and more complex models. With that said, it is clear that, for example, there could be a lot of interesting computations happening at the level of dendrites. Single neurons can do amazing computations. For example, they can multiply 2 inputs, they can integrate inputs in nonlinear ways. And a lot of the basic models that we have been discussing and will continue to discuss may be instantiated at the level of dendritic computation. So one of the most basic ways of thinking about single neurons and how they compute is the notion that there are inputs that come from the dendrites from other neurons, and those are integrated, and in a very simple circuit that contains a capacitance and a resistor. And after integration of those currents, there's a threshold, and based on that threshold, the neuron either it's a spike. Yes.
I have a question on the last slide. With these different models, like it seems like one way to test how useful they are to actually explain is just like look at variance explained with the different models. Have people like looked at that and shown like how useful it is to use a way more complex one versus a more non-compartmentalized one?
Right. So I think to ask how useful a model is or not, I think we need to ask. We need to discuss this in the context of specific question. So we'll come back to this discussion later when we talk about networks. There are some, by definition, if you're interested in looking at the nonlinearities that happen at the level of dendrites in integrated information, by definition, this model doesn't have that, and we need to get into this type of models.
Like firing rate of the output neuron. Like that seems to be like.
So in several cases, so if you inject current in the soma of a neuron and you want to look at the output firing rate, this may suffice, and I think I have a slide to show that this type of very simple single compartment models can do extremely well in describing that aspect. So again, it depends a lot on the question. I'm not trying to argue that one should never go this way. Again, depending on what exactly you want to do with the model, this may become less or more shown. I want to postpone the discussion. I want to come back later when we talk about networks. We'll talk about arguably one of the largest computational efforts these days in neuroscience in Europe, the so-called Blue Brain project, where people are trying to build really large-scale models of circuits of neurons at this type of resolution. So they really have multiple compartment models with a lot of biological details. And at that point, I'll open the discussion of how useful they are or not. If the only question you care about is trying to predict the firing rate of a neuron given a certain current injection, these models do reasonably well. So just to make sure that we're all on the same page, when people model a circuit and the components are integrated and fire neurons, they typically use an equation like this, which describes this very simple RC circuit. Now it works. Okay. So this describes the I is the current that's injected into the neuron, and that current goes into a leak component through this resistance, and is also integrated through this capacitance. So this is a very simple differential equation. I'm not going to go through the code. It's a very simple implementation. And basically what happens here is that the current is integrated, voltage increases depending on this input current. And when the voltage reaches a threshold, then this neuron emits a spike. Most people also institute a refractory period, meaning that the neuron cannot fire immediately after fires a spike. It's very simple, it's pretty fast, and it captures some of our essential intuitions about neurons and how they integrate information. It certainly does not consider a lot of important biophysical phenomena, including spike rate adaptation, multiple compartments. It doesn't really explain the sub-millisecond shape of action potentials, and it doesn't include the neuronal geometry. So again, I'm not going to go through the code. This is not a perfect implementation of an integrated and fired neuron, but just as a very simple way of instituting these with the Euler integration method. Essentially, in one line in MATLAB, one can actually simulate what's happening here. So this is one of the main ways in which people think about single neurons and put them in the context of more complex. So as I was saying before in response to a question, if you have these are actual data. These are recordings from in CAD in primary visual cortex. What happens when you inject a given amount of current and this is the firing rate that you get. So these are the recordings that you get in context, and this is what you get from this integrate and fire model. And this is what you the line is the integrate and fire model. The points here correspond to the first two spikes. So the first two spikes emitted by this neuron in response to current injection are well approximated by this very simple model. If you look at all the spikes, one of the things that happens is that there's adaptation. The neuron starts to fire more slowly over time, and that's one of the things that's not well captured by this integrate and fire. So one notch higher in complexity in terms of modeling individual neurons is the Hodgkin and Huxley model. And this goes back to the very basic equivalent electrical circuit that I described before, where we have a variety of different ionic currents that flow through the neuron. And one is trying to describe the injection of current in terms of the integration, as well as the current that passes through each of those different membrane components. So these funny numbers that you see here, this N, M, H, and all these funny exponents that you see here, capture the shape of the action potential, and were actually predicted by fitting experimental data by Hodgkin and Huxley way before the advent of molecular biology techniques to actually describe the exact nature of all of these channels. This is a relatively simple model still. It's more complicated and more time-consuming in terms of the computational cost than integrate and fire neurons, but it can describe very well the detailed shape of the action potential in response to the depolarization and hyperpolarization caused by the flow of sodium and potassium ions. So again, these are simulations. Again, I'm not going to go through the code in any detail, but one can do experiments and record the voltage and clamp voltage for a particular neuron at different levels, and then measure the conductances for potassium and sodium, and show that those can be described reasonably well by this simple Hodgkin-Huxley type of model. So now I'd like to switch to describing and giving you an overview of different ways in which people study brains, and give you a flavor for the type of possibilities and challenges that we have in terms of trying to ask questions about computations at the level of brains and neurons. So people use a variety of different techniques to study brains, and those are roughly illustrated here, indicating the spatial resolution and the temporal resolution, ranging from studying synapses all the way to studying whole brains. So some of these techniques you may have heard of or you may be familiar with. It's possible to patch clamp, meaning to be able to fix the voltage and study individual channels within a neuron. So that's really a very, very high spatial and temporal resolution. At the other extreme, there are techniques like scalp EEG and scalp MEG. These have very high temporal resolution. They give you information about brain function at the millisecond level, but very coarse spatial resolution. So this is capturing activity at the level of several centimeters of cortex. So this is averaging activity over large samples of neurons. Then there are techniques, non-invasive ways of interrogating brain function, like PET or functional magnetic resonance imaging, fMRI. And these are techniques that have the advantage of A, being non-invasive, B, being exhaustive in the sense that one can study, at least in principle, entire brains. But they have very poor spatial resolution as well as temporal resolution. These are local field potentials, which can be obtained by inserting electrodes into the brain and have relatively coarse spatial resolution but very high temporal resolution. And then perhaps one of the gold standards in terms of studying neural circuits is the examination of the activity of individual neurons, which can be obtained by recording extracellularly from high impedance microwires. So this is a scale of several tens to hundreds of microns, and one can again get at the millisecond dynamics of wiring in circuits. And I'll give you some examples of what can be inferred from several of these different techniques. So the very before really getting inside neurons and circuits of neurons, as I already alluded to at the beginning, one of the major types of insights that are often derived about function of specific brain circuits comes from studying lesions. And this is one example that many of you may have heard about. There's a particular part of the brain, typically encompassing the temporal lobe and more specifically fusiform gyrus, where damage to this area renders people unable to detect phases. This is known by the name of prosopagnosia. And it's a pretty striking effect. Oliver Sacks, the famous writer and neurologist, once wrote a book entitled "The Man Who Mistook His Wife for a Hat." So these are people who have really a major deficit that's highly specific. They can recognize all types of objects, but they cannot really identify faces. And this gives you an idea of the distribution of lesion sites in cases of people who have prosopagnosia. So yes, this is localized, it's not the entire brain, but it's not a single specific location. There's a wide variety of locations that can lead to this type of deficit, in part because these are not really experiments that are designed, these are accidents that happen either due to carbon monoxide poisoning or due to a virus infection. But there's a wide variety of areas, more or less circumscribed to the areas around the fusiform gyrus that lead to the major deficit in face detection. Perhaps somewhat less known is another example. This comes from studying people who have lesions in the parietal cortex. This is the case of people who have chemi-neglect, meaning that they can see and they can recognize shapes very well, but they have a major deficit in attending to or visual recognition in one half of the visual hemisphere. So this is what happens when this is an example from one subject that had hemi-neglect, and this is when he was asked to copy different types of drawings, and typically these people will copy essentially one half of the drawing and basically completely ignore the other half. So yes, they can recognize the object, but they basically focus only on one field and not the other. And as I also alluded to earlier, one of the remarkable aspects of computations in the brain, as opposed to computers and circuits, is the remarkable ability of the brain to adapt and recover from major lesions. So this is an artist that had hemineglect, and this is his self-portrait 2 months after he had a stroke. And this is his self-portrait 3.5 months later, and this is his self-portrait 9 months later. So when he had hemineglect, he was essentially drawing very coarsely only half of his face, and then essentially he recovered, in spite of the fact that this person had major damage in part of the right parietal. Another major way to study brain function, of course, comes from the behavioral studies of different visual illusions. And you probably have seen many of these. And just to focus on one of them for the sake of discussion, you probably have seen this type of visual illusions where this green circle seems to be much smaller than this one, in spite of the fact that if you take out all the context, all these blue circles here, they are exactly identical. So one might imagine that if we have a really accurate description of computational models that can actually describe brain function accurately, it may also make these type of mistakes, and it may also be able to explain a lot of these different visual illusions. So the performance of computational models in some of these visual illusions is sometimes taken to indicate the extent to which these computational models are doing things in a way that's similar or not to human. There's also a very famous one, the Margaret Thatcher illusion. It's very hard usually to recognize faces when they are upside down, when they are in the right orientation. It's actually much easier. And to a first approximation, I would argue that a lot of the computational models that we'll talk about today do not actually have this type of face inversion illusion. This is not to say that we have to reproduce every single visual illusion in computational circuitry, but this may point us to some constraints in terms of the type of computations that are implemented in Okay, so one of the main techniques to investigate cortical function involves recording the activity of neurons. And this is a schematic from David Hubel showing an electrode in the extracellular space of a neuron. And by using high impedance penetrating microwires that are advanced into different parts of the brain, it's possible to capture the action potentials that the neurons are firing. So if you record extracellular data from an electrode like this, you get a signal that looks more or less like this. This is the voltage as a function of time. And then investigators typically will high-pass filter this signal to get a signal that looks like this, which contains all of these all or none spiking events. And then another signal that looks like this that contains the so-called local field potentials. Depending on how close this electrode is to the neuron, one can get single unit activity, that is record from a single neuron, or as in this case, what we refer to as multi-unit activity. This probably means that there are several different neurons that may contribute to the signal. There may be one neuron here, and there may be another. And there are techniques that people use, usually referred to as spike sorting, to try to disambiguate this type of recordings and be able to disentangle and separate the different neurons that contribute to this signal. This local field potential signal is less well understood in terms of the biophysics. I argued earlier that to understand the biophysics of the generation of action potentials relatively well and model that very well with the Hodgkin-Huxley equations. There are a lot of people trying to understand the biophysics that underlie the generation of local field potentials, but that's not as rigorously understood as the case of spikes. Yet this type of signals may also be quite informative in terms of understanding the representation of information, not by a single neuron, but at the level of a cluster of neurons. So just to give you a couple of examples of the type of recordings that people have done, the type of information that can be gathered from listening to the activity of neurons. This is the work of Bob Desimon when he was working with Charlie Gross many years ago at Princeton and subsequently also and previously also at MIT. And essentially by chance, they landed on the notion that there are neurons in higher parts of visual cortex that respond in a very selective fashion upon presenting pictures of faces. So Bob Desimon, by the way, will be here on Saturday, and he will tell us more about his work. In this particular case, what they were doing was recording from a neuron in inferior temporal cortex. And this is a typical way in which investigators represent the activity of the neuron response to a given picture. So they show a picture of a hand, in this case, and then the x-axis here represents time, and the y-axis here represents the number of spikes per second. So it's a scale here. So this neuron fired vigorously in response to this picture, for example, and less vigorously in response to this picture. So this is an example of the type of recordings that people do to try to interrogate how neurons and circuits respond to different shapes. This is another example. This is a neuron that responded to macaque monkey faces predominantly, more so than to other stimuli. More so, the neuron responded to particular viewpoints of the face with respect to other stimuli. And as I said, this was largely discovered by chance. We still lack a systematic understanding of what exactly drives neurons in higher parts of visual cortex. In particular, at the time that these recordings were done, Bob Desimon and Charlie Gross were studying neurons in inferior temporal cortex. And a few years earlier, they had come up with the discovery that neurons respond to complex shapes such as faces. After a very frustrating day when they were using very simple stimuli such as lines, inspired by the work of people like Hubel and Diesel, who had shown that neurons respond to simple contours and oriented lines. And they discovered that the neuron was one of the neurons they were recording from was particularly would fire particularly vigorously in response to whenever the investigators passed in front of the monkey, and that gave rise to a whole industry or a whole set of studies that concern what exactly is driving these neurons that continues to this. So we know that there are neurons that respond to these type of complex shapes, and we can interrogate some of the basic properties of the circuitry by this type of. Another technique that people use to study brain function involves electrical stimulation. So it's possible to inject current both in humans as well as in monkeys, and then behaviorally evaluate what the effect of those currents is. So here's the classical work that William Penfield did. William Penfield was a neurosurgeon working with patients that have epilepsy, and as part of the procedure to treat epilepsy, they would insert electrodes into different parts of cortex and inject current and behaviorally evaluate what the subject, what effect of current. They did this in a wide variety of different places in cortex. And here I'm showing just one example of what has come to be known as the homunculus, meaning the idea that there is a particular strip in the somatosensory cortex that represents different parts of the body. meaning that if he stimulated, when he didn't feel stimulated in one of those areas, people reported having a sensation in that particular area. So there's a map of the entire body along the somatosensory cortex that was elucidated by electrically stimulating different parts of it. In connection to the previous slide where I showed you that there are neurons in inferior temporal cortex that respond to complex shapes such as faces, this is a recent example of work doing stimulation where Fras and Felix were stimulating an inferior temporal cortex, the same area that Bob Decimal and Charlie Gross were studying that I showed in the previous slide. And they were asking whether, upon recording from a neuron that responds more vigorously to faces than to other stimuli, what would happen if you inject current in the vicinity of that neuron. Mind you, when you're stimulating, both in this case as well as in this case, you're not really stimulating the activity of a single neuron, you're stimulating the activity of a large cluster of neurons. So here I'm showing the psychometric behavioral curve, showing how many times the monkey reports seeing a face as a function of the amount of visual signal. So they used stimuli that range from largely faces with minimal amount of noise, faces with more noise, signals that contain absolutely no information about faces, and then signals that contain information about other objects. And that's what's represented in this axis here. So as you go to the right on this figure, you should be able to discriminate faces better. And indeed, in the absence of stimulation, the monkey here reports most of the time that he's seeing a face. As you go to the other extreme, this picture here, and this part of the diagram here, the monkey reports that he almost never sees a face because it's pretty easy. In particular, in the middle, the monkey should be approximately a chance. When they show this picture, the monkey reports 50% of the time they see a face and 50% of the time that they don't. This is a two alternative force choice. Give me 1 second and I'll answer your question. So here, upon electrical stimulation, that is upon injecting current within a cluster of neurons that responds to faces, the investigators showed that they could bias the monkey's performance, that is, shift monkey's performance a little bit towards in the direction of reporting faces more often than under the control conditions. Yes?
So you're saying that when there was no information at all, he was more likely to say that there is a face, and then when there was information about another kind of object?
So let's focus on this condition here. This is the 0% condition. Here's when there's no information at all. So if I ask you, is there a face or not, you would say, I don't know. But if I force you to say yes or no, in this case, monkeys are reporting whether it's a face or not at chance. Basically, this is this point here. If you look at the blue curve, which is when there's no stimulation, the monkey is reporting 50% of the time that they see a face, 50% of the time that they don't. They have to report something. They cannot just say, I don't know, there's nothing there. Okay, so they're forced to either say yes or no. Okay? When they are stimulated, when you inject current via this electrode, this is what happens. So there's a small, according to the author, significant increase in the number of times that the monkey is reporting seeing face. So in this particular case, about 60% of the time, the monkey says, I see a face, even though there's absolutely nothing there, presumably as a response to the subjective sensation elicited by injecting current in a part of the brain that's responsible for recognizing faces.
I don't understand why does he say 50% of the time that he sees a face if there is no information at all?
Why is 50 as opposed to?
Less than 50.
As opposed to 0?
I mean, I expect that 0 would be in some kind of 50% noise level.
So to say 0, so here they have to say it's a face or it's not a face. So 0 would mean that they're always saying it's not a face. So here 0 would be that they're always saying it's a flower. Okay?
So it's face or flower, not face or absence.
Well, it's not face or absence. It's face or something else. That's what the monks are saying.
Opposite, not face.
So they cannot say nothing. So in the other case, the alternative is in this particular case is flower. It's not always a flower. It's always another stimulus that's not a phase. Okay. But so here 0 would mean that here they always see a flower, for example. So that's okay.
And the reason they'd want to say things is to get a reward if they get it right, and it's just going to randomly assign face or not face from a non-coherent spot. If I remember the study correctly.
Say it again. Yes.
If I remember this particular experiment correctly, for the 0% visual signal, they just randomly assign whether or not they'll get a reward if they guess face for that.
Correct. So the question is, so monkeys have to be trained to do this type of task. So the question is how they are rewarded. So exactly how you reward is critical in terms of what monkeys do. But the essential point here is that there's no information, and they shouldn't. If they're away from 50, that means that they have a bias. And in this case, they're not. Okay. So I'd like to now give a brief preview of some of the different techniques and the different insights that people in Thrust 2 in the study who are studying neuroscience are doing as part of the CVMM, and how this can inform several aspects of computation. So I will start by very quickly describing the work of Windrich, Freivold. Again, Windrich will be here on Saturday to give a full presentation. So Windrich has followed on this tradition of examining different parts of the temporal lobe of the inferior temporal cortex in monkeys. And what's particularly exciting about his work is that he's combining two of the techniques that I mentioned. One is functional imaging, and the other one is recordings via electrodes. And what Windrich can do is start by looking at a brain in a very coarse way with functional imaging, and thus discover particular parts of the brain that are responsible for discriminating faces from non-faces, but then target that particular area with electrodes. So this is a coarse to fine approach, if you want. You first identify an area that's involved in a particular task of interest. In this particular case, in the context of seeing and recognizing faces in a transformation in variant way. So he can identify many of these, what he calls patches of cortex that are involved in discriminating faces from non-faces. And then he can target those areas with electrodes, and then record the properties of those neurons, and, for example, show that there are some patches that are important in discriminating faces, but they do not have tolerance to the specific viewpoint. That is, that they respond specifically to one viewpoint of the face, similar to the neuron that I showed you before by both Tessimon and Charlie Ross. Whereas they may be more advanced parts of the visual hierarchy, where there are neurons that respond to different faces in a transformation tolerant way. If the neuron can respond to all of these different versions of the face, in contrast to this one. So the combination of functional imaging and single neuron recordings can be extremely powerful, because it allows us to take complex tasks where you would initially may not have any clear idea of where to stick electrodes to investigate the neural circuit properties. So you can pinpoint the essential aspects of the essential geography of where those computations are happening, and then go in with electrodes to really try to uncover the mechanisms and try to ask detailed questions about potential algorithms that are involved in that computation. As I said, Windrich will give us a full presentation of that line of work on Saturday. This is work from my own group, and we also investigate physiologically what happens inside the brain, but instead of working with monkeys, we work with humans. And the way we can interrogate invasively the human brain is by collaborating with neurosurgeons who work with patients, typically epileptic patients, that have seizures, and where the seizures cannot be treated by pharmacological means. And in that case, the neurosurgeons typically will implant electrodes for two reasons. One is to mark where the seizures are coming from, and also to be able to basically functionally determine the properties of different parts of cortex in order to try to resect the part of the brain that's responsible for the seizures. Because the location of the seizures is not known a priori, the neurosurgeons end up inserting a large number of electrodes in different parts of the brain, only a small fraction of which ends up in the epileptogenic areas. This gives us the opportunity to interrogate the human brain in the context of complex cognitive tasks at a very high spatial and temporal resolution. Depending on the patient, we often record field potential signals such as the ones that I'm showing here. This is a recording from what's called an ECOG electrode, a 2 millimeter low impedance electrode that records an intracranial field potential signal as a function of time. And here each color corresponds to pictures from a different object category. And this is showing a more vigorous response in this electrode for in the blue curve to pictures that contain faces. And again, we looked at the invariance of the response to different scales as well as different viewpoints. So this is a response that happens in the inferior temporal gyrus, and where this electrode showed selectivity, meaning that it responded more vigorously to some pictures compared to others, and it did so in a way that's largely invariant to this type of stimulus manipulation. This is another example. This is a case where we recorded from the amygdala using microwires in a case where we could obtain the action potential responses. And each of these pictures was shown to the patient. They were shown in random order. The patient was discriminating the presence or absence of the face. And each of the ticks here that you cannot see very well represents an action potential from that neuron. There are multiple repetitions of each picture. And these posthumous histograms are shown in the same format that I showed you before for the macaque monkey data. The response of the neuron, the y-axis is the firing rate as a function of time. So this is a neuron that had a more vigorous response in response to these three different pictures. And what's quite remarkable about this is the degree of selectivity. The neuron did not just respond to any face, it responded to this particular person, but also the large degree of invariance in response. These pictures are as different as you can imagine in terms of color, size, contrast, and many other properties. And yet this neuron responded to these three different pictures that included former President Bill Clinton. So by interrogating the activity of circuits in the human brain, we can start to get some insights into basic computations that can give rise to visual selectivity and visual preference. Arguably, one of the major revolutions in neuroscience over the last several years is the work of people like Ed Boyden, who developed what's called techniques that are often referred to as optogenetics, meaning that it's possible to target specific types of neurons by virtue of using the specificity of molecular biology, and in that way be able to either stimulate or silence specific types of circuits within neural circuits. And this is work that Bob Desimon did in collaboration with Ed Boyden. I'm not going to go into the details again. We'll hear a full presentation from both of them on Saturday. But the notion here is that by using light and a particular photosensitive molecule that's controlled by a promoter that's expressed in specific types of neurons, one can shed light onto the circuit and then activate a specific type of neurons, or in another type of experiment, silence specific subcircuits. So this is illustrated here in a cartoonish type of way. You have a circuitry with lots of neurons that become activated, and then by using light, one can control a specific subset of neurons that are being silenced. And there's the number of studies using this type of technique is increasing on a daily basis. And the reason I'm very excited about this is that this can provide us with the opportunity to really take computational models and really test them at unprecedented resolution, but also take biological data and try to infer what are the main algorithms that are involved in computation. So back to the questions about visual recognition, one can imagine that we can start to dissect the type of circuitry that's involved in generating selectivity and invariance. Another thing that Ed Boyden is doing is developing a new set of high-density multi-electrode arrays that we hope to use across different species to interrogate the activity of large numbers of. One of the limitations that we have in neuroscience is that we can only interrogate small chunks of circuits at a time. So many of you may wonder how far can we really go by looking at one neuron at a time. So this may give us the opportunity to record from hundreds, if not thousands, of neurons simultaneously. Ed Boyden is a fantastic neurotechnology inventor and a very creative guy. He's far more grandiose than I am, and he likes to say that we're going to be able to record from 100,000 neurons at a time. And I hope that will happen, and maybe some of you will lead the revolution and go to that. But even if we were able to record from 1,000 or 100 or several hundreds of neurons simultaneously, I think that can play a transformative role in terms of how we can dissect algorithms from neural function.
I just have a terminological question. Does interrogate mean something specific?
What I mean is record voltage. Record voltage as a function of a millisecond precision extracellularly, meaning that we can really get the activity of neurons at high temporal resolution. So one more, two more examples, and again we're going to hear a full presentation on this on Saturday. This comes from the beautiful work of Matt Wilson, who's recording the activity of neurons in the hippocampus as well as many other areas to try to understand spatial navigation in rats. So this pertains to the question of perhaps trying to answer one of the CDMM challenges, where am I? How can we get information about location from the activity of a set of neurons? So Matt Wilson has pioneered the description of hippocampus function at the level of ensembles of neurons and decoding the activity of an ensemble to be able to describe the position of a rat as it's navigating in the environment. And more recently also, he's been doing a lot of interesting work trying to look at the interactions between different areas in the context of spatial navigation. This here aims to illustrate work done by Bob Desimon in collaboration with Tommy Boggio, where they were looking at the question of what's there, that is how to decode information about a set of objects. And one of the projects in Trust 2 has to do with decoding activity from a population of neurons or from field potential data to describe a set of objects. And in particular here, they were interested in decoding information from an ensemble of neurons in areas before and inferior temporal cortex, with in the presence or absence of attention. That is, when the monkey is or is not paying attention to the stimuli. So here the basic setup is that they are reporting from a set of neurons. In this particular case, they are putting together neurons recorded in different sessions. We usually refer to this as a pseudo-population. This is not a real population of simultaneously recorded neurons, but rather a set of individual recordings that are then concatenated. And then we have an ensemble of many dimensions, meaning multiple neurons, and a set of labels for which picture was presented. And we typically use machine learning techniques similar to the ones that Lorenzo described a few days ago to try to decode the activity of this neuronal population. And the basic finding here is that what we can decode the activity of a population of neurons when they are when objects are presented in isolation. B, when the objects are not attended, the decoding performance is significantly lower. This is a measure of the decoding performance as a function of time. And when an object is attended to, the performance of this decoding procedure is much higher. So attention is a major way of filtering information that can significantly enhance the possibility of decoding information from a population of neurons. So to bring this back to the main goal of trying to understand computations that pertain to intelligence, and to take as an example one that is particularly dear to my heart, which has to do with visual recognition. What can we learn from studying neurons and circuits of neurons? And I just gave you a very brief tour through a variety of different responses that one can obtain from cortex. So why do we want to pick inside the brain, and what can we learn from recordings in the brain about these type of computations? So one of the reasons why I'm optimistic about the notion that studying brains can inform our endeavors to look at computational algorithms that solve intelligent problems is that more recently we have amazing resolution to get at wiring diagrams and the connections between neurons that were not available ever before. This is done at low resolution in humans by using non-invasive imaging techniques such as diffusion tensor imaging in combination with functional imaging that give us sort of the lay of the land in terms of the interactions between different parts of cortex. And more recently, there's a whole set of people that are very excited about the notion of what people refer to as connectomics, and that is trying to understand connections in the circuit at a very increasing resolution. And this is by no means the answer. It's not that once we have a wiring diagram, we'll be able to immediately infer computations and how we see and recognize and how Messi plays soccer so well and so on. But it will provide a set of constraints for our computational models that were not available before. It will really guide our thinking about architectures and computations in ways that were not possible a few years ago. The other aspect that I think is rapidly changing and why it's, I think, exciting to be young and this field now, I'm referring to you, not to me, is the strength in numbers. So there's rapid progress in the ability to record the activity of large numbers of neurons, and we have a series of mathematical tools to be able to try to probe and model these type of responses. So I would say that the last 3 or 4 decades we have been painfully studying one neuron at a time or a few neurons at a time. But there are major aspects of computations that will be elucidated over the next several years by being able to study many neurons simultaneously. And finally, I like the notion of being able to look at the source code. And by that I mean the possibility of manipulating neural circuits, and this will happen particularly in rodents and macaques, to be able to examine necessary and sufficient computational elements. So this alludes to the work that I very briefly described from that was pioneered by Ed Boyden, where we can really target specific types of neurons, and people are using this more and more in a variety of different systems. So in the same way, for those of you who speak computer science, if I give you a binary code and I ask you to figure out what it's doing, you can try different inputs, different outputs, and you can try to mutate or change different binary, but it's not that trivial. In opposition to that, if I give you the source code, you should be able to read that in much easier format. So here we have an opportunity to debug the system, to be able to control the system and manipulate the system in ways that were completely unheard of a decade ago.